everybody. Sorry, slight mix-up. I thought I was chairing in the afternoon, but it's the morning. So now we're having the members' platforms. We've got three people to present. The first is Samia, who I think we heard a little bit about yesterday in one of our talks, talking about attenuated heart-brain integration predicting functional non-epileptic seizures.
So good morning, everyone. Thank you very much for the opportunity to present this research, and thank you to the organisers for putting together a wonderful programme. So my name is Samia. I'm a neurology registrar doing a PhD in South London, and I'm going to be talking to you about functional non-epileptic seizures. So just some introductory points. So functional non-epileptic seizures are also known by lots of other names, including dissociative seizures, non-epileptic attacks, and also psychogenic non-epileptic seizures. But these are essentially paroxysmal events which are characterized by alterations in motor, sensory, autonomic, and/or cognitive functions. And they can often resemble epileptic seizures, but are not due to epileptiform activity. And these are very common, so particularly if you work in neuropsychiatry or epilepsy, and up to 30% of patients referred to us with refractory seizures actually don't have epilepsy and they have functional seizures. There's often quite a significant diagnostic delay, and in that time patients can receive inappropriate medications with anti-epileptic drugs and also anaesthetic agents. And they're also associated with a high morbidity and mortality, and importantly, quite a sizable socioeconomic cost to both patients and the healthcare service. So I think there are two main problems. I think the first is that the diagnosis can be quite tricky, particularly if you're in clinic and you're relying upon a history. You might not always have a video. And the second is that we don't fully understand the mechanisms which underpin functional seizures. So in terms of the mechanisms, this has sort of been work that's been led by probably several of the people in this room, but they include dissociation and more recently interoception. And as we heard from our wonderful speakers yesterday afternoon, interoception can be described as this perception of the internal bodily state. So as it gets closer to lunchtime, you'll probably be feeling quite hungry, and I'm feeling quite nervous because I'm on this platform and I can feel my heart beating. But in people with functional seizures, this process is impaired, so they're less interoceptive than healthy controls, and the interoceptive deficits are also associated with higher seizure frequency and increased levels of dissociation. And we can measure interoception at sort of a variety of levels, as Professor Garfinkel discussed yesterday. And commonly, this is measured at the behavioral level, so for example, through doing heartbeat tracking tasks. But we can also measure interoception at the level of the afferent signal. By that, I mean really assessing the relationship between the body, in this case the heart, and the brain. And we can measure the sort of neural representation of this afferent signal in terms of something called the heartbeat evoked potential. So the heartbeat evoked potential is an event-related potential which you can measure on the EEG. And this represents a synchronized neural response to individual heartbeats and is therefore described as a marker of interoceptive representation. And interestingly, in people with depersonalization, derealization disorder, the amplitude of this signal has been shown to be reduced. So could we use a dynamic marker like the heartbeat evoked potential to try and look at what happens to people before their functional seizures? And could this give us some sort of insights into the mechanisms behind them? So that was exactly what we wanted to do. So the aim was really to assess whether alterations in interoceptive processing measured through the HEP occur before functional seizures, and to compare this to the most common differential diagnosis, which is epileptic seizures. And we hypothesized that there would be a change in the amplitude of the HEP from the interictal, so between seizure state, to the preictal, so just before a seizure, in those with functional seizures, but not in those with epilepsy. And could this give us some mechanistic insights into what happens before functional seizures, and could this also be diagnostically useful? Okay, so to do this, I used clinical video telemetry data, and just in case any of you don't know what this is a rather strange test where we bring patients into hospital, or sometimes we can do the test at home, and we connect them to EEG, ECG, and also video. And the aim is to try and capture and video an attack, and then see what's happening in the brain and also the heart at the same time. So from this, I basically tried to identify the event onset, and that was based on the clinical video and the final neurophysiology report. And I then clipped out sections of pre-ictal data, so this is the period of time just before a seizure, and also the interictal state, so this is away from seizures and free from any epileptiform activity. And essentially the idea was to look at the HEP in these two states and also between our two clinical groups. So the EEG was de-identified and exported, pre-processed, and the heartbeat evoked potential analysed. And we had 25 people with functional seizures and 19 people with epilepsy. And just in terms of the pre-processing, this was based on previously published methods. So the EEG was re-referenced common average, filtered and downsampled. As I mentioned, the heartbeat evoked potential is an event-related potential which is measured in relation to the R wave of the ECG. So we used a ECG software to detect the R peaks and used independent component analysis to correct for large artifacts such as blink and muscle. And we focused on the frontal and central electrodes because these are the areas where the HEP is thought to be maximal. And we took a number of measures to exclude confounding cardiac effects. So what did we find? So these plots basically show you the HEP waveform, and I'll just talk you through what this shows. So on the y-axis, we have the HEP amplitude, which is measured in microvolts. And on the x-axis, we have the time in seconds, and this is time measured in relation to the R peak, which is at 0 here. And we're measuring the HEP in this time window in the shaded grey box, which is just zoomed in on the bottom. And the green lines represent the interictal HEP, so away from seizure periods, and the red lines represent the pre-ictal HEP, so just before a seizure. And what you can see, if you look at the functional non-epileptic group to start off with, is that this pre-ictal HEP, so the red line, is much lower than the interictal HEP. And this was significantly different with an effect size of 0.6. Whereas in the epilepsy group, we actually don't see this at all, and there's no difference between the HEP in the two states. And if anything, the pre-ictal HEP is slightly higher than the interictal. And you can also visualize this in a slightly different way. I'll just stand here so you can see. So the functional group are the dark circles, and the epilepsy group are the clear circles. And essentially in the functional group, you can see this reduction in the amplitude of the HEP between the interictal and pre-ictal states, whereas we see the opposite thing in the epilepsy group. And we see the same thing at F8. So again, in the functional group, there's this significant reduction in the amplitude of the heartbeat evoked potential between the interictal and the pre-ictal state, but we don't see this in the epilepsy group. So again, this is demonstrated here. You can see this significant reduction between the interictal and pre-ictal state in the functional group, whereas the epilepsy group don't really change. So what I've shown you so far is just looking at between states, but within the same clinical group. But we also wanted to compare both of the groups directly. So to do this, I calculated something called the HEP amplitude difference, and this was quite a crude measure. It was just basically the interictal HEP subtracted from the pre-ictal HEP. And this gave us a composite measure which we could directly compare between the two groups. So if you look here, you can see the HEP amplitude difference at C4 and at F8 is significantly different between the two groups. And finally, we want to see, well, could this be diagnostically useful? So in order to do that, I looked at something called, calculating the receiver operating characteristic curve. And this was basically the HEP amplitude difference plotted across all frontal and central channels, and also including sex into the model. and I haven't mentioned here, but we had a greater proportion of females compared to males in the functional group, as you might expect. So when you look at the data in this way, this has an area under the curve of 0.893 and a sensitivity and specificity of 0.84 for differentiating epileptic and functional seizures in our sample of patients. But I will obviously just say that there's never going to be one magic test for differentiating between these two groups. Okay, so in conclusion, in those with functional seizures, we see a significant reduction in the amplitude of the HEP between the interictal and the preictal state. We don't see this in the epileptic seizure group. And this suggests that aberrant interoception, or this sort of abnormal communication between the heart and the brain, might underpin disembodiment, which occurs before functional seizures. And this change in HEP amplitude might therefore reflect a neurophysiological biomarker of functional seizures, and a positive biomarker at that, because we don't see this in the group of people with epilepsy. There are several limitations, so this was a retrospective study, and we also excluded several cases to avoid any confounding cardiac artifact with the HEP calculation. So I'd just like to thank you all very much for listening. I'd also like to thank my PhD supervisors, Mark and Mahinder, who's here as well, the funding body, and also this list of collaborators, particularly David, who's a very talented neurophysiologist at George's. So thank you for listening.
Okay, thank you so much, Samia. I think we have time for a few more questions on that fascinating talk. Do we have a microphone? Does anyone have a microphone? No. We'll start with a question from over here, this gentleman.
I can shout.
I think we probably should wait for a microphone for the audience at home. Oh. Here you are.
Thank you.
Thanks.
Were all the epileptic seizures the same? Because I know that we see that there is an important minority of epileptic cases where there are significant changes in heart rate preceding seizures. So I wondered if there were subgroups within that.
Yeah, thank you for the very good questions. So most of the patients who we had in this study were people with actual temporal lobe epilepsy. But in terms of your question, because of the confounding cardiac effects and the influence that can have on the heartbeat evoked potential, we actually excluded cases where there was a significant difference in heart rate between the two states. So that actually meant we had to chuck out quite a few patients, but that was just sort of because we wanted to make sure the sample was as clean as possible. I think it's really important to look at this work in those group of patients, as you say, so that they're not excluded from the sample.
Thank you. And Tony David?
Well done, really interesting study. You may have said this, but do you know what are the mechanisms for attenuating the hair? I mean, do we know whether certain drugs will do this, other physiological conditions? I mean, I like your interpretation of what it might mean, but can we try and figure out what the mechanism might be?
Yeah, so from sort of my reading of the literature, and I'm sure some of the interoceptive experts can comment as well, my understanding is that this is recognised as sort of a marker of interoception, and that is because it's sort of a neural representation of the heartbeat. But I think there are also other things which can influence the HEP that are described in the literature. So things like attention and pain. I don't know about effective medications actually, but I know there are other factors that can sort of influence it as well that are important to recognise. Thank you.
Okay, we have time for one very quick question from John Stone.
Hi. Great study. It's a similar sort of question, a bit more specific, because a lot of data on functional seizures points to it being a bit of a sort of cousin of panic attacks in terms of the sort of pathophysiology, but obviously with important differences. Do we know, and obviously people do dissociate during panic as well, do we know what you might find in a HEP study in people with panic attacks?
So I don't actually know what you might sort of see in that. I expect with those with panic attacks, you might also get sort of a preatrial tachycardia, and you might also get effective respiration, and whether that could influence the heartbeat evoked potential, I'm not too sure. The dissociation which you mentioned is also really important because it would be interesting to sort of correlate measures of dissociation with this as well, but I'm not sure what you'd see with just panic attacks. Sorry, I don't know.
Thank you. Well, we're going to have to move on to the next speaker now, so thank you so much to Samia Elkommos.